Hello and welcome. Today's interview is with Alexandra Klein, who works with a group of researchers at the Max Planck Institute of Neurobiology, studying how the brain and the body regulate emotions. Alexandra shared with me what she's learned from some neat experiments studying fear responses in mice. In particular, she gives evidence for the role of two structures in the body that have become rather trendy in recent years: the vagus nerve and the insular cortex. I hope you enjoy the insights that Alexandra brings, and please remember to subscribe to the In a Sense YouTube channel for more information about how your bodily signals impact your health and well-being. Thank you very much. Hello, Alexandra, and thank you very much for taking the time to speak to us today on the In a Sense channel.
I'm happy to.
And I'd like to start really by hearing a little bit about your background. I saw your research paper published or introduced on Twitter, and that helped me realize that you were studying interoception. But can we have a little bit of a background about your career and how you ended up working at the Max Planck Institute of Neurobiology, studying how the brain and the body work together to regulate emotions?
Yeah, of course. So, I did, after school, I studied biology first because I actually wanted to do psychology, because I always wanted to understand how the human brain works and how, in general, people work. But then I noticed, okay, psychology, like the actual job, is not what I actually want to do. So I wanted to more understand the basics of how the brain works. And so I did biology in my bachelor in Munich. And then for my master's, I went to Berlin to do more medical neuroscience. And that was also quite interesting. But that was very focused on working with humans, which still didn't give me the answers to how the brain actually works. And so I went back to Munich to do my PhD at the Max Planck Institute for Neurobiology, as you said, and joined there the group of Nadine Gogola. And so her group is actually called Circuits for Emotions. And so we're really interested in understand how emotions work and how emotions are implemented in the brain. And that really fascinated me. And because I never really thought of this so much, but then reading about her research was like, yeah, that's actually what I wanted to do and what I want to understand. And so we are working on, trying to understand the brain circuits for emotions and working on mice. And so I could really dive more into the question on how actually the brain works. And how I ended up in this brain body project was also more or less a coincidence, because my project was more revolving around the topic of fear and how fear can be regulated. And then I just by accident found a connection between how body and brain interact to regulate fear. And this made me super excited in general about interoception, self-awareness, and how body and brain work together to regulate emotions.
It's nice that your team are able to focus quite specifically on that topic of circuits in the brain that regulate emotion. I'm hoping that we can talk a little bit about that experiment that you've just mentioned in a while. But first of all, maybe you could help us understand where some of the gaps are in research at the moment. So what are the main gaps in our understanding as a scientific community in general about how the brain processes information from the body to regulate emotion?
Yeah, so there are, of course, things already known and how body and brain interact. Most of this knowledge comes from human studies and from, I guess you also know that, from very classical behavioral studies where participants, for example, are asked to count their heartbeats, for example. This is a very classical task to assess interoceptive abilities. And yeah, so it works well and people can report that. And then usually the studies try to also correlate some brain regions with that by putting people in functional MRI scanners. And then they see roughly brain regions like lighting up that are active during these tasks. And from that, we kind of have a rough knowledge of which brain regions are involved in these processes. But since this is mostly from these human studies, really like it's not much known how it actually works. So where are the connections? Like where are the signals that come from the heart actually are transmitted to the brain, and how the brain exactly has this ability to also send down again information to the body to really regulate the emotions. And yeah, so like these exact brain regions and connections and what's the actual signal that is transmitted there, we don't know much yet. It's just more rough ideas, I would say.
Yeah, and that's it's surprising. I've only got into this area of research relatively recently, and as you say, it's rough ideas, and I think that's from an outsider, I thought so much more has been figured out than actually is figured out.
No, sorry when I interrupt, but it's actually true, because especially in the terms of emotion research, it's like the first ideas of when people wanted to understand emotions were that actually, the body plays a huge role in how emotions are actually happening. And like back in the earliest 20th century, the idea was first the body has a reaction, and then the brain only knows, my body reacts, or my heart is pounding, or my palms are sweaty. This must be an emotion. And then that was the ideas in the early 20th century, and it was forgotten somehow. And people were then so much focusing only on the brain until only a few decades ago, when people were like, actually there is a strong connection. But beforehand, it was largely just ignored.
Yes. Yeah. And I don't know about your experience, but I remember as an undergraduate learning about the autonomic nervous system. So it's almost like we had a lot of the different components there in biology, but they weren't linked together to, psychology or emotion research.
Yeah, absolutely.
What's one, I'm really excited to have you on the channel, because you are the first researcher that has come onto this channel who studies emotion regulation in mice. And which, looking down the list of papers that you and your group have published, I see that a really interesting title that caught my attention was a paper on facial expressions in mice. And obviously, as humans, we have this idea that our emotions are expressed through our face, but I had no idea that mice have facial expressions linked to emotions. So could you please explain a little bit about that study? I understand it involved machine learning, and explain what you learned.
Yeah, no, absolutely. I mean, we're all on one hand surprised that it actually worked, but on the other hand, we were working so much with mice, and just looking at them, it was somehow for us always clear that they do express something in their faces. But it was never really shown in detail. There were some studies already, for example, in rats that showed if they're, for example, in pain, they have some painful facial expression, but it was always over a very long time and more used for animal caretakers, basically. And so we, especially my colleague, the first author of the paper, he was then very interested in actually showing that mice do express their emotions in their face, just as humans do, which is very intuitive. And so, what he did is that he basically took very close-up videos from mice when they were, for example, experiencing pleasure. So when they were getting a drop of sugar water, or like a slight painful stimulus to their tail, and different stimuli like this. And then using machine learning and some complicated algorithms, he actually found that these facial expressions are very stereotypic. That means, so like each mouse has similar facial expressions, and that they're very specific to the type of stimulus and the underlying emotion. And this was very consistent and surprising, but also on the other hand, like, yeah, makes sense that also mice do have those facial expressions.
People who've maybe they've had pets at home, intuitively sort of know when something has changed for that animal, but from a kind of more how we view animals as a whole as being able to communicate their emotions, that's is quite surprising in that way. Mm-hmm. So what expressing an emotional state is one thing. So these mice, when they have different stimuli, whether it's something pleasurable like sugar, then they express an equivalent of a smile, let's say. So the machine algorithm could detect those patterns, but how about communication between mice?
Yeah, very good question. So because in our minds, in human minds, of course, we only have those expressions as a social communication tool, right? Mm-hmm. In humans, that's absolutely clear, right? So we use facial expressions to express emotions, to trying to evoke the same or similar emotions in others. In mice, we don't know this yet. We know for sure that, we know for sure what seems very likely is that facial expressions also have another important function. For example, it is to like orient the sensory organs towards a stimulus, for example, when they like it, or away from the stimulus when they don't like it. And we really see if there's a drop of sugar, the whiskers go in front, the ears go in front. And so basically to absorb everything that is there. Mm-hmm. But for the question, if they use it for communication, we don't know yet. But we're trying to understand that and working on that, because it's, of course, super exciting and interesting to just do this experiment, just put two mice in front of each other and see if the facial expression of one animal changes the facial expression of the other animal. We're not there yet, but I think it's a very super interesting and also quite easy experiment. So I maybe can tell you soon.
Oh, okay. Yeah. Look forward to hearing the outcome of that if it's work in progress. Mm-hmm. The other question that's come to mind about that is that, so there's differing opinions, I'm sure you're aware, amongst emotion expression in humans about whether there's this universality or not. So, and it appears that, or many researchers argue that there isn't it's more cultural rather than universal expression, and that would be so fascinating to know in mice as well, or other creatures, about whether there's a more reflexive, as you said, just move towards the stimulus, or whether there's possibly some cultural or learned expression as well.
Exactly. I mean, as you say, it's still debated, and researchers are not really don't have this one opinion about that. And I think looking at facial expressions of emotion in animals is definitely a very promising way to go, because I doubt that animals can really influence their facial expressions intuitively, and just I try to smile even though I actually feel pain. So I think that would really lead more to the actual roots of facial expressions and what they're used for. So
good. And I'd like to in a minute move on to your recent paper about fear, which is something that's really important to everyone in their everyday lives. And so in this experiment, you were modeling fear in mice. And before we go on to that, I'd like to sort of just allow you to explain about the ethics procedure for working with mice. It's many years ago, I was involved in some work in with animals, rodents, and I'm aware that there's a process. But for some of the people watching this video who may have concerns about working with animals and doing experiments that induce fear, if you could explain how this works in a university setting.
Yeah, no, absolutely. And I'm very thankful that you also give me the chance and the time to explain that, because I know, of course, it's a sensitive topic, and I'm happy to speak openly about that. Because, so it's like animal ethics are different in every country, but and even in every state in Germany, it's slightly different. And I'm in Bavaria, and Bavaria is definitely one of the most strict countries, which I agree, because it has to be controlled. And so the procedure is that if we plan experiments, we have to write a animal license protocol, and this has to be then decided about in the government. And so within these protocols, we really have to explain in detail what we want to do, how many mice we intend to use, and the goal of it is always to reduce animal numbers and, of course, to limit any suffering to the minimum, and to have like meaningful outcomes from all the experiments. And it's usually quite a long and very strict procedure before you can even start doing any experiments. And I totally agree, because I absolutely it has to be controlled, because we're working with animals, and so I'm always aware of what we're doing there. But I also, on the other hand, see the necessity of, especially in our field, if I want to understand how brain and body interact, it's not doable in a dish with cells. And that's why I'm very happy and thankful that we still can do these experiments using animal models.
That's a really nice overview. And I remember I've still got a friend I'm in touch with on Facebook, actually. She was one of the caregivers at the animal house that we worked with. And she is such a big animal lover. And it's, that was what I really liked, that she'd chosen to work in the animal houses because she loved animals so much. So I do see that there is often a lot of care given to these. Yeah. So with that in mind, for your recent paper, how did you model and measure fear regulation in mice?
Yeah. So, of course, the best would be to just be able to ask a mouse, how do you feel today? But of course, a mouse can't report us or just by using words or anything. So what we have to do is to use behavioral experiments. So we try to, by looking at the behavior of mice, to infer their underlying emotional state. And so fear is I guess, the most studied emotions in mice, because mice tend to be fearful animals. I guess everybody who's seen a mouse in the house knows that it would just immediately run away. And we kind of make use of that. And there are different behavioral experiments, how you can test how anxious a mouse is. And in my case, what I did in my experiments is that I wanted to first teach a mouse that it has to be scared of a certain stimulus. So in my case, it was a tone that the mouse was scared of. And this is very classic. So this is also used in clinics, actually.
Sorry to interrupt. So a tone, is it just like a loud beep or?
Exactly. So it's not only the tone. So how I taught the mouse to be scared of the tone is that I showed the tone several times. And every time at the end of the tone, it was coupled to a mild, painful shock to the mouse. So a tiny electrical stimulation to the paws, so that it learns over time that when the tone comes, it predicts that this small painful stimulus would come. And so that over time, when the mouse was hearing only the tone, it was scared of only the tone, basically.
A bit like the Pavlov dog experiments. But
it's very classical Pavlovian conditioning, exactly as you say. And so this process is called fear conditioning in the laboratories. And it's one of the most highly used experimental paradigms in mice when you want to study emotions, because then you basically know that there's one stimulus that the mouse is afraid of. And you can really, it's specifically that one stimulus that you then can test. And in my case, I just I did not only want to teach the mouse to be scared, I wanted to know how the brain can actually then regulate this newly learned fear. And to do that, the second experiment that is happening basically after fear conditioning, I wanted to teach the mouse to be not scared of the tone anymore. And this is a process that's called fear extinction. And this is really also used in clinics, for example, for war veterans, when they're scared of some, for example, helicopter sound, that they're just exposed to the sound over and over and over again, so that over time they learn, okay, it's actually not scary anymore. And I did exactly the same with the mice. So again, I showed the tone several times, and this time without any like shock or any painful stimulus at the end, so that the mice over time were supposed to learn that they don't have to be scared of the tone anymore. And I wanted to see how the brain and the body then interact to actually achieve this like regulation of fear. Because importantly, you would think it's healthy if I don't have fear at all, and if I immediately forget about fear, right? That's the intuition. But on the other hand, you also have to remember that fear is also a very essential emotion that keeps us from doing stupid stuff, for example. And so you want to somehow achieve a good balance between not being so scared that you can't move or can't go outside the door anymore, but also keep your like healthy fear of things that are really dangerous. And so this balance is very essential.
So I imagine in a population of mice, just like in humans, there's a variation in how afraid people become, but also how quickly they recover or extinguish that fear. So what did you discover in the mice? And I was interested, from the little bit that I read about this, you were looking at the freezing aspect as well as how much they freeze.
Yeah, exactly. So as you say already, the readout that we use to assess how scared a mouse is that we look at their freezing behavior. And also humans show that when they're are scared, and then this shock freeze, right? And you don't move anymore, and your body is just in pure, like, no movement whatsoever. And this is the same in mice. So mice also have this fear reaction. And so I measured how scared a mouse was by measuring how much it was freezing when I was presenting the tones. And interestingly, I found that there seems to be, as you say, a huge spread of mice, and some are very fearful and some are actually not fearful at all. And I've done many, many experiments, and this just seems to be that there is a population, and it's like in humans, some are very scared of the same stimulus and some are just not. So and it seems to be the same in mice. Yeah.
And so there is this difference in how scared they are, and then what about the how quickly that fades away? Does that vary?
Yeah, so that's what I then found when I did the fear extinction experiments. So interestingly and unexpectedly, I found that the mice, like this huge population of mice, like the super scared and the not scared ones, they somehow manage all to extinguish to a similar degree. So if like the brain is intact and if they're healthy, then there seems to be a mechanism in the brain that just makes them extinguish very equally. So that very fearful mice, they extinguish quite fast, and mice that are not as fearful, they're somehow not extinguishing that fast, so they just all end up some in the middle at this fear balance, at this healthy fear balance that we actually want to keep to survive.
So you don't see any, and you said you've lots of experiments have shown this. So in humans, we have conditions like PTSD or phobias where, from the way you've described it, I would understand that as the extinguishing phase doesn't. The people in the population, they don't all level down together. Some people, it carries on for a long time. So you don't really observe that in mice at all.
I don't see that in my mice, but I think the big difference, which also is like the one big part of the study that I did, because in people suffering from these types of anxiety disorders, there it has been shown that they have some brain areas that are either hyper or hyper, so too active or too little active. And this can explain a lot of these observations that have been done in those people suffering from these disorders. Because what I found, because I wanted to find the brain correlates of what is actually happening there, and the brain region that I was mostly interested in, if I can already start explaining that,
Yes.
is the insular cortex. And the insular cortex is, for me, one of the most interesting brain areas because it has been shown, as I just mentioned, it's involved in many different types of psychiatric disorders, for example, anxiety disorders, and also people suffering from traumatic disorders. And this has been well established in humans. And for me, the most interesting bit is that it is also known as the interoceptive cortex or brain area. So a lot of signals that originate from within the body, they actually arrive in the insular cortex. And we know that these signals are processed there and can also, again, the brain area can also in turn give signals back down to the body to also regulate then reactions in the body. And this is all, again, from human studies. We don't know exactly how it works. It's all very correlative. So I wanted to look at the insular cortex exactly in my experiments. And so I did that. And what I did is that I switched off the insular cortex while the mice were undergoing this fear extinction training. And then suddenly, very interestingly, I found this huge spreads in behavior, so that animals that were very fearful in the beginning, when I take out the insular cortex activity there, they extinguish almost not at all. So they keep being fearful. In comparison with the mice that are not very fearful, when I take out insular cortex activity there, they extinguish in a second and they forget their fear super fast.
Wow. So can you say that again, please? So the two groups, the really fearful ones, if you take out the insular cortex, then
they keep being fearful. They don't forget their fear.
So it lasts longer, whereas the ones who are not very fearful, it shortens.
It shortens and like super quickly. So they immediately unlearn to be fearful very, very quickly.
And at this point, just remind me in the experiment, so they're no longer getting the electric shock, they're just getting the tone at this point.
Exactly. They just get the tone. And so they're only supposed to, let's say, unlearn their fear of the tone in this experiment. And this unlearning goes very, very slow in the high fearful mice and very quick in the low fearful mice.
So what do you have any hypothesis about the mechanism for that? So if you take out the insular cortex, what's happening for those two different populations? Do you have any clue?
Yeah, I mean, it also leads to the rest of my study a bit, but the actual mechanism is really hard to understand. So how I imagine it, very roughly, is that we need insular cortex activity and normal, physiological activity that is then used for to inform other brain areas that are then actually doing this unlearning, let's say, because we know the insular cortex has connections to a lot of those brain areas that have already before been shown to support this unlearning of fear. And somehow this physiological activity in the insula is needed to actually talk to this downstream areas that then actually say, okay, in a less fearful mouse, it's supposed to unlearn a bit, slower in comparison to these high fearful mice. What the actual cellular mechanism is, we don't know yet. This we don't know, but we definitely know that the body plays a very important role in that.
So that is really fascinating. So it really that your experiment really does show or indicate that the insular cortex, whatever the tendency that those groups of mice have, whether they're overly fearful or less fearful, the insular cortex brings back that unlearning to a kind of hopefully a useful level, and when it's taken away. So does that can you sort of maybe comment on how this can inform what we already know about the insular cortex and in humans, and also maybe about how we may be able to use this information? I know it's very early days, but for use again in humans for keeping emotions within a safe range therapeutically.
Yeah, so absolutely. I mean, so before I reply to exact that question, maybe I also add this tiny bit that's also very important to know, to show how important it actually was what we found out there. Because what we found is that when mice show this freezing behavior, it's very classical fear behavior, we found that interestingly, their heart rate is also dropping. So always when a mouse started to show this freezing behavior, heart rate was also going very quickly, very down. Oh, I hope my internet is stable.
Yes. Yep.
Yeah. Okay. Good.
Yeah.
Great. Because sometimes it's a bit weird. And so we found that the heart rate is dropping, and even more interestingly, we found that also in the same time frame when the mouse starts to freeze, also the insular cortex activity seems to have a drop, and this was very tightly correlated. And so then it was kind of logic to us that, well, the mice that are highly fearful, they freeze more, right? And they have more often this drop in heart rate activity and also this drop in insular cortex activity. And so we found, okay, somehow this must be the key to understand the differences in this high and low fear population, and how it actually can lead to this intermediate balanced fear levels. And that was super interesting for us. And then we thought we need to understand just this tiny bit more if it's really the heart rate. And what we did is that we then interfered with the communication between the heart and the brain. And this happened via the vagus nerve. So vagus nerve, maybe, I guess, also people who know interoception, they know about the vagus nerve, I guess. Mm-hmm. And it basically brings most of the information from the body to the brain, and also vice versa. And when we just interrupted this communication via the vagus nerve, then we saw exactly the same as when in the experiments when we took out insular cortex activity. So high fearful mice stay fearful, low fearful mice extinguish very quickly. And so that actually was the thing that made me really believe, okay, it's actually very relevant what I found there, because vagus nerve stimulation has been in clinic already, and it's currently even tested for treating anxiety disorders. And also what I found extremely fascinating, the idea that just by controlling your body, so for example, the heart rate, you can control your fear level. Mm. And this might be some external route, where we just can control some body reactions, where we don't even have to go and target the vagus nerve, but just change our behavior, change our emotions, and regulate our emotions. And that I find super fascinating.
Mm. Yes, indeed. So it sounds like you've really been able to piece together some of the pieces of the jigsaw in this study. And as you said, vagus nerve stimulation is in the UK here. Through the National Health Service, you can get a little zapper device, vagus nerve stimulation, just to battery powered to go on the neck, and it's used for, it's licensed for a number of different things, I believe, for migraines as well as anxiety and so on. This really is adding credibility to lots of different approaches, really. And as you said, if we can, things which regulate some basic physiological functions like heart rate or breathing. That adds to the evidence of how profoundly these may work for mood or mental health conditions in general. So even something like listening to music that lowers the heart rate.
Yeah, exactly. And I mean, also, as you say, when you also had yoga experts, I think it all makes sense, because I mean, to influence heart rate is not that easy, I guess, because I guess you can by like more meditating and, as you say, maybe also listening to music. But I also found it fascinating that breathing, for example, is also very easy for us to influence. And I think really working on breathing can actually really have strong influences on the body and on the brain, and vice versa then.
Mm-hmm. And maybe, what you've learned about the insular cortex as well. So if there are scanning people to look for individual differences, if someone's got an underdeveloped or underactive insular cortex, then that may help understand why they don't respond so well to something like breathing. Or if the vagus nerve, if there's a disruption, it's helping to see at what stage there may be a dysfunction.
Exactly. Yeah, I think that makes it very relevant, because especially with this experiments with vagus nerve stimulation, I thought also this can be very informative for the clinics in the end, because we for the first time showed that it does not only have beneficial results, because I also saw that in these mice that have these high anxiety phenotypes, or these high fear mice, that it actually is not beneficial. It just like keeps them being fearful when we interrupt this signaling. So I think, it has a lot of potential for clinics and for human research. That has to be kept in mind there.
This is also such good research. I'm really going to keep an eye on your research. But before we close, I'd like to find out whether, outside of your research, what you've learned about emotion regulation and interoception. Do you have any personal experiences to share about internal body awareness and its role in your own life?
Yeah, I mean, actually, yes, because very classically with the first lockdown, as I guess 50% of the people worldwide, I started doing yoga. And I have to say this like internal focus, breath work, and like really focusing on my internal state helped me a lot with my work, with focusing, with like basically saying what is important, what is not, and with just improving my everyday life and my health somehow. I'm not an expert practitioner, for sure not, but I really feel just the benefits of spending half an hour a day with like really looking into the state of my body and to really acknowledge that. I think that's the very few perks of the lockdown. But I'm happy that I started doing that.
So it gave you time to focus inwards and to understand your own body. That's brilliant. I'm super grateful that you and other researchers are you're pioneering some of this work and also walking the talk and really using these techniques on yourself. It's and that's why we need science because there's lots of people who've done maybe they've been to yoga or had some successful therapy or a therapist themselves, and they've all discovered maybe something through their own body, but to then have the science to back this up, I think it's an exciting time for the whole the therapeutic world.
Yeah, it's true. I absolutely agree. I mean, there's still a lot to discover or to be discovered, but we're on a good path. And I think in general, I feel interoception research is really on a rise right now. And many people are very interested in actually understanding what is there and what is the connection between body and brain. So I guess there's lots more to come in the future.
There is so many more questions popping up all the time. Yeah. So what visions do you have for your research? Where do you hope it will get in the next maybe 5 or 10 years?
Mm-hmm. So for my personal research, you mean, or in general, like this area?
Maybe both, whichever.
Yeah. So well, I personally am very excited that this was my PhD work. So I'm gonna now go for a postdoc. I'm gonna go to the States, to a lab at UCSF, and I'm very excited. And I definitely continue on body-brain interactions and their role in emotions, looking more at the breathing aspect of the body. I'm very excited to do that. And I really feel that this is also where the research in this area is going now, to really understand how the brain, or like what comes first, like is it really the body, is it the brain, is it the interaction? It's still not known, huge debates, and I feel the answer is somewhere in between. Mm-hmm. And I really feel that this is where the research right now is going, that we actually try to understand the circuits and the exact brain regions and connections that do this interoception, and then really try to use that information to really know, okay, if we can influence the physiology of the body in a certain way, we have the tools to help people suffering from diverse diseases. And we don't have to have drugs acting or not necessarily, or super invasive surgeries or something like this, but that we actually use this knowledge that we create to help ourselves somehow. And I don't think it sounds esoteric, or I hope not, but I really feel that there is such a huge potential that we don't know yet, that we hopefully can understand in the near future.
I agree with you. It maybe a couple of decades ago, it would have sounded esoteric, but
now we know the science behind it, so it's not.
What the science does is it gives a language for all these other therapies that already exist to come together and say, okay, this is what I was seeing. This is what I was seeing from this angle, and here's the common understanding.
Exactly, yeah. And I'm happy to be at this connection. Very exciting.
I really appreciate the time you've given us today. Is there anything else you'd like to say before we close?
Well, I want to say thank you. And I'm really happy about this opportunity to speak about my research, because, we don't get to do this a lot. It's mostly speaking through the publication. So I'm very grateful that you gave me the time also to speak. So thank you.
Thank you. And where can people find out more about your work?
So of course on Twitter, I think that's the easiest. So my handle is Axolovsky with AE in the beginning. So I guess this is hopefully findable. And I'm happy about anybody reaching out, having questions or wants to discuss. So that would be nice.
Thank you. And I'll add your Twitter handle to the description of this video.
Mm-hmm. Thank you.
Thank you very much, Alexandra, and I hope to stay in touch.
Thanks. Me as well. Thank you.